This is a lecture on the accessory organs of the digestive system. This lecture is based on Martini's chapter 25. Learning objectives, drawing assignments, and let's begin. So, digestive system, salivary glands. There are three pairs of salivary glands: the submandibular gland, the parotid gland, and the sublingual gland. They secrete saliva, which is 99.4% water, and some mucin, a slippery glycoprotein. Remember, glyco means like sugar, carbohydrate type protein, and salivary amylase. Salivary amylase digests carbohydrates. The submandibular glands are in the floor of the oral cavity. They secrete 70% of saliva. In the picture to the right, they are sort of teal color. The parotid salivary glands lie over the masseter muscle. They secrete 25% of saliva. The sublingual salivary glands are found under the tongue. They secrete around 5% of saliva. Salivary gland secretion is increased by parasympathetic nervous system and decreased by the sympathetic nervous system. Having food in the mouth stimulates a salivary reflex with increased saliva secretion. Okay, now the liver, an overview. The liver is the largest visceral organ. It weighs around 3 pounds. It is intraperitoneal. It has 4 lobes: the right lobe, the left lobe, the small caudate lobe, and small quadrate lobe. Around 33% of the blood supply to the liver is from the abdominal aorta to the celiac trunk to the common hepatic artery to the hepatic artery proper. This blood has high oxygen content. Around 66% of the blood supply to the liver is from the hepatic portal vein. This blood has high nutrients and a low oxygen content. Tributaries that drain into the hepatic portal vein include the superior mesenteric vein, which drains the small and large intestines, the inferior mesenteric vein that drains the large intestine, the gastric veins from the stomach, the splenic vein from the spleen, and the cystic veins from the gallbladder. Venous blood leaves the liver through the hepatic veins that drain into the inferior vena cava and then the heart. The blood has high nutrients and a low oxygen content. The blood has high nutrients because this blood has all of the nutrients that were processed by the liver after they were received by the hepatic portal vein. And it has low oxygen content because the hepatic portal vein that entered the liver had a low oxygen content. Only the blood that entered the liver and the hepatic artery proper had high oxygen content, but the liver used up the oxygen in the functioning of the liver. So now this blood has a low oxygen content. The liver is innervated by the autonomic nervous system, and the function of the liver is also controlled by hormones. You can look at the picture here showing you the sympathetic nervous system and the parasympathetic nervous system. Connective tissue structures. The liver has a tough fibrous capsule deep to its visceral peritoneum. Otherwise, the liver, which is rather mushy, would not be held together by just the visceral peritoneum. So there's a tough fibrous capsule that holds it together, and then there's the visceral peritoneum. The coronary ligament attaches the liver to the diaphragm. The falciform ligament attaches the liver to the anterior wall of the abdomen and the diaphragm. The round ligament contains the remains of the umbilical vein. And the falciform ligament separates the liver into a right lobe and a left lobe. Liver lobules. The liver is divided into around 100,000 lobules. Lobules are columns with a hexagonal shape. The hepatocytes in the lobules are arranged in rows, like the spokes of a wheel around a central vein. Take a look at this picture, and now take a look at this picture here that's showing only a part of the liver lobule, but gives you more of a sense of the fact that it has height to it, let's say, and it's not just a little flat structure. We'll get back to the little zoom in window in a bit. Okay, portal triads. A portal triad is a branch of the hepatic portal vein, a branch of the hepatic artery proper, and a bile duct grouped together at an outer corner of a lobule. Each lobule has 6 portal triads at the 6 outer corners. So here's our hexagon. So here would be a portal triad, portal triad, portal triad, portal triad, portal triad, portal triad. The blood from the branches of the hepatic portal vein and hepatic artery proper enter the lobules. The blood that enters the lobules drains into the central vein, which are in the center. The branches of the hepatic portal vein branch into many tiny sinusoids that pass between the hepatocytes. These sinusoids are capillaries with many tiny holes or fenestrations in their cell membranes and gaps between the endothelial cells in their walls, in the walls of the capillary. We haven't done blood vessels yet, so we'll review this when we get to blood vessels. And there's a picture in the right lower corner of the slide. And what you're looking at here is a capillary that's been cut in half. And if you look closely at the capillary, you will see that there are many, many tiny little holes, and those holes actually go through the cell membrane of the endothelial cell. And then this picture doesn't show it, but one single endothelial cell and its neighbor would have unusually large gap between the two endothelial cells instead of the endothelial cells being closely linked together. There is a perisinusoidal space between the sinusoid walls and the hepatocytes. The blood in the sinusoids flows into this space and bathes the hepatocytes. The hepatocytes absorb nutrients from the blood and secrete substances into the blood. The hepatocytes have microvilli on their surfaces to increase their surface area for this absorption and secretion. And here's the zoom in. And in the center of the zoom in is the sinusoid space with the blue endothelial cells on the outside. Note that the endothelial cells have fenestrations. Fenestra, is a Latin word for windows. So when they say they have fenestrations, it's a fancy way of saying they have little holes, but they're saying that the cells have little windows in them. And here then we see this tiny space between the sinusoid and the rows of the hepatocytes with their microvilli. And this is called the perisinusoidal space. It used to be called the space of Dis, but we're trying to get away from eponyms. The branches of the hepatic artery proper branch into capillaries that pass between the rows of the hepatocytes. The blood from the capillaries brings high oxygen to the hepatocytes. The capillaries join the sinusoids and both empty into the central vein. And I have a nice red arrow here showing you the direction of blood flow from the corner of the hexagon of the liver lobule, and the blood flow is going into the central vein. The blood in the central veins flows into hepatic veins. There's a right hepatic vein and a left hepatic vein here in the picture, and then the inferior vena cava. So the key thing to remember is that when you're talking about the hepatic portal vein, you always have to put the word portal in, because if you just say hepatic vein, then you're talking about these veins that drain the liver into the inferior vena cava. So hepatic portal, the word portal is the distinguishing word. Hepatic portal vein brings blood from the intestines into the liver, and then the hepatic veins collect up all of the blood and bring it to the inferior vena cava. Hepatocytes produce bile. Bile makes fats easier to digest and carries waste into the intestines for elimination. Bile flows into tiny bile canaliculi, shown here in green, that empty into the bile ducts in the portal triads. And the flow of bile, therefore, going in the opposite direction from the flow of blood, which is going towards the central vein. Here we have bile, which is going away from the direction of the central vein. I always thought that was especially notable that you had the flow of blood going in one direction and the flow of bile going in the other direction. That's a key point in studying the lobules. So try to learn that. The bile ducts lead to the right and the left hepatic ducts. From there, the bile can enter the duodenum directly from the common hepatic duct and then the common bile duct. Alternatively, the bile can pass through the common hepatic duct, then go into the cystic duct and enter the gallbladder for storage. Just a little detail there. I think that the words have to really be sorted in your mind for these different ducts, because right and left hepatic ducts don't have the word bile in them, which would have been nice, like if it was right and left hepatic bile ducts. But so just kind of silently put the word in there in your mind. And then the common hepatic duct, same thing, common hepatic bile duct, you can put it in your mind. And then the common bile duct. So, the right and the left hepatic secret bile duct are draining the little bile ducts that are at the corners of the liver lobules in the portal triads. And then the bile is collecting into the common hepatic bile duct. Once the cystic duct has come off of the common hepatic duct, the name of the duct past the cystic duct changes and becomes the common bile duct. Okay, and here are some pictures of liver lobules. This is a histology slide on the left. However, this is not a human liver lobule shown here. The human liver lobules do not have such a clear connective tissue boundary, so they used the liver lobule of another creature. I don't know which ones. They just say it was a mammal, just to show you the histology slide with a somewhat clear liver lobule. And then here is a zoom in on a portal triad showing you that the branch of the hepatic portal vein is large and central. Then we have to the left a tiny bile duct here, and the branch of the hepatic artery proper is seen to the right of the branch of the hepatic portal vein. So moving on to liver functions. Overall, the liver has around 200 functions. All of the nutrients that have been absorbed in the small intestine and large intestine are in the venous blood that drains into the hepatic portal vein. The liver processes the nutrients in the blood before the blood drains into the hepatic veins, the inferior vena cava, and the systemic circuit. So here's a list of important liver functions. 1, it processes and stores nutrients. 2, it produces plasma proteins. 3, it produces bile. 4, it removes waste and toxins. 5, it removes old red blood cells and pathogens. And that is not the only place that old red blood cells and pathogens are removed. It is just, an important place. Severe damage to the liver is life-threatening. Okay, processing of nutrients. That picture to the right is not to be learned, but to show you that the liver is doing a lot of processing of nutrients. So the liver monitors the level of nutrient molecules in the blood, including carbohydrates, amino acids, and lipids. And it has incoming blood from the hepatic portal vein. And it performs many chemical reactions that change nutrient molecules in the incoming blood into different nutrient molecules as needed by the body. It therefore maintains normal glucose, amino acid, and lipid levels in the blood. And it also makes cholesterol. And I have a tiny little slide to the left showing you the many things that cholesterol does. I think that because we're so used to cholesterol being measured as a sign of ill health, the person has high cholesterol, it's dangerous. But actually, cholesterol is needed for cell membrane homeostasis. So it's actually quite important. And you can look at all of the other functions that cholesterol has. Okay, so continuing with liver functions, the liver stores nutrients. Excess nutrients after it has received blood from the hepatic portal vein and it's done its transformations of nutrients into nutrients that are needed by the body. If there are excess nutrients, they are stored in the liver. Excess nutrients are stored in the liver as glycogen and lipids. Glycogen is a storage form of glucose. The liver stores fat-soluble vitamins, which are vitamin A, D, E, and K. It stores iron from the breakdown of old red blood cells that it is doing. It can store the nutrients as needed. The liver also stores blood as a part of the venous reserve. And venous reserve is just blood that's in the vein part rather than the artery part of our cardiovascular system, our systemic arteries and veins. There happens to be more blood in the venous part than the arterial part. And that blood can be used if there's ever blood loss. Then the blood that is hiding in different organs, such as in the liver, can be used as reserve blood. So that's what we're talking about when we say the liver stores blood as a part of the venous reserve. For example, the spleen also stores blood as part of the venous reserve. So the liver is only doing part of that job. Okay, so the liver also produces plasma proteins. Plasma proteins include albumin, globulins, and fibrinogen. Albumin is a plasma protein, meaning a protein that is carried around in plasma, and plasma is the water part of blood as opposed to, red blood cells or platelets. So albumin, this protein, helps hold plasma in vessels by osmotic pressure. It's like a little sponge that's holding water in vessels. And albumin transports substances like unconjugated bilirubin, a breakdown product of hemoglobin. When red blood cells are broken down, hemoglobin inside them has to be broken down. And one of the breakdown products is bilirubin. It's called unconjugated when it hasn't been conjugated yet, but it's a form that is insoluble in plasma. So it needs to be held by something that is soluble in plasma that can, be carried through plasma. So it's held by albumin, which then transports it in the blood and actually carries the unconjugated bilirubin to the liver, which conjugates it and turns it into conjugated bilirubin. Albumin is very important, both because it holds plasma in vessels and also because of its job transporting substances. It transports many substances other than unconjugated bilirubin also. Globulins also contribute to osmotic pressure in vessels and also transport substances in the blood. Fibrinogen is a different kind of protein. Fibrinogen can be converted to fibrin that helps form blood clots. The liver also makes angiotensinogen, which is a hormone that helps maintain blood pressure and volume. And we will learn more about fibrinogen when we do blood vessels, and we will learn more about angiotensinogen when we learn about the urinary system. I know you're thinking, what does the angiotensinogen have to do with the urinary system? Well, with the kidneys, which produce a hormone that helps to maintain blood pressure and volume, it's part of the system that starts in the kidney with certain hormones. And then the liver does its part by contributing angiotensinogen. Other organs of the body are involved, and there's a whole system that we'll learn about that helps to maintain blood pressure and volume. But for now, if you can remember, kind of put a pin in it, the liver makes angiotensinogen, and that will come up again. The liver makes bile, and bile is a mixture of water, bilirubin from the breakdown of red blood cells and hemoglobin, phospholipids, cholesterol, and bile salts. Bile is secreted into the duodenum, and bile breaks up or emulsifies ingested fats. Okay, so you'll often hear the word emulsifies. It means breaks up. Bile makes the fats easier for lipase, which is secreted by the pancreas, to digest. Bile carries waste products that are eliminated in the feces. The liver breaks down wastes that are in the blood. It breaks down hormones. It metabolizes toxins like alcohol and drugs like aspirin and many other drugs, many other toxins besides alcohol. And lobules have resident macrophages that patrol the sinusoids. And you see here in purple, there's a resident macrophage there patrolling, sort of rolling around in the sinusoids, and the macrophages will engulf damaged red blood cells and any pathogens that have gotten into the liver, most likely through the hepatic portal vein, and they would be in the hepatic portal vein because it would be in the blood that was being drained from the intestines. That might be a place that pathogens would get into our blood, by crossing through with perhaps a nutrient that the small intestine was absorbing, and with it might come a bacteria or a piece of a bacteria, and it might get into the liver and then be engulfed by the resident macrophages. Okay, moving on to the gallbladder.